Hello and welcome to The Sleepy Scientist. Before settling in for tonight's quiet journey into biology, please take a moment to like the video and subscribe to the channel if gentle explorations of science are something you enjoy drifting off to. It helps this sleepy little corner of the internet continue quietly growing. And while you're getting comfortable, it would be lovely to know where in the world you're listening from tonight. Perhaps a quiet bedroom somewhere after a long day, a dimly lit living room with a blanket and a cup of tea, or maybe headphones on during a late-night train ride home. Wherever you are, thank you for being here. There is something rather fitting about that question, actually. Because wherever this video is being heard tonight, something extraordinary is happening inside every single listener. Inside every cell of the body, tiny molecules are quietly following instructions that were written long before any of us were born. Those instructions are written in DNA, a molecule so small it can only be seen with powerful microscopes. Yet it somehow contains the information needed to build bones, muscles, eyes, skin, nerves, and an entire human brain capable of wondering how any of this works in the first place. And that raises a rather strange question. How does DNA know how to build you? After all, DNA is not alive in the way cells are alive. It does not think. It does not plan. It does not understand the difference between an eye and a finger, or between a heart and liver. And yet, from a single microscopic cell, a complete human body unfolds with remarkable precision. Cells divide, tissues form, organs emerge in the right places, complex systems develop, communicating through chemical signals and electrical pulses. Trillions of cells somehow coordinate their behavior with astonishing reliability, all guided, in some sense, by sequences of 4 chemical letters stored in a molecule coiled tightly inside the nucleus of every cell. It can feel almost as if DNA contains a kind of knowledge, a hidden instruction manual quietly directing the construction of life. But of course, molecules do not know things. They follow the rules of chemistry and physics. So the real question becomes even more intriguing. If DNA does not know anything at all, how can it build something as complex as a human being? Tonight, we will slowly explore that question. Not as a single simple answer, but as a long story stretching from the earliest discoveries of cells and genes through the molecular machinery of proteins into the strange world of developmental biology, where embryos organize themselves with remarkable precision. Along the way, we will meet the scientists who uncovered these mysteries, explore the hidden networks of genes that coordinate development, and gently untangle the processes that allow a fertilized cell to become a living, breathing organism. Because the truth is both simpler and far more astonishing than it first appears. DNA does not know how to build you, and yet through billions of years of evolution, chemistry has learned how to do something extraordinary. It has learned how to build life. Inside the body, hidden within nearly every cell, there exists a molecule so small that even powerful microscopes struggle to reveal its full structure. Yet this tiny molecule carries something extraordinary. It contains the information required to guide the construction of an entire human being. Bones, muscles, nerves, blood vessels, skin, eyes, and brain cells all emerge through processes that begin with this microscopic thread of chemistry. That molecule is called deoxyribonucleic acid, more comfortably known as DNA, and it raises one of the most quietly astonishing questions in all of science. How does a molecule know how to build a human body? At first glance, the idea seems almost absurd. Molecules do not think. They do not understand anatomy. They have no awareness of organs or tissues. They simply follow the rules of chemistry and physics, bumping into one another and reacting according to the forces that govern atoms. And yet the instructions contained within DNA guide the development of organisms with remarkable consistency. A fertilized human egg begins as a single cell smaller than the width of a grain of sand. From that single starting point, cell division begins. One cell becomes two, two become four, four become eight. Soon there are thousands, then millions, and eventually trillions. Each of those cells contains nearly identical DNA, and yet those cells do not all become the same thing. Some cells develop into neurons capable of transmitting electrical signals across vast networks inside the brain. Others become muscle cells that contract and relax in coordinated rhythm. Still others form skin, liver, bone, blood, or the delicate tissues of the eye. All of these specialized cell types emerge from the same genetic instructions. The scale of this coordination is almost difficult to comprehend. The human body contains roughly 37 trillion cells. That is 37,000 billion microscopic units of life, cooperating with extraordinary reliability. Each one performs specific tasks, communicates with neighbors, and contributes to the larger structure of tissues and organs. Despite the complexity, the process usually unfolds with astonishing precision. Cells divide when they should divide. Structures form where they should form. Systems connect in ways that allow the body to function as a single integrated organism, all guided in some sense by DNA. To understand how scientists began uncovering this mystery, it helps to step back several centuries to a time when the existence of cells themselves had not yet been discovered. In the mid-17th century, an English natural philosopher named Robert Hooke examined thin slices of cork under a primitive microscope. The instrument was little more than a collection of lenses mounted in a brass frame, but it revealed something remarkable. Hooke saw that cork contained tiny box-like compartments arranged in a repeating pattern. They reminded him of the small rooms occupied by monks in monasteries, known as cells, and so the term cell was born. Hooke did not realize that he was looking at the remains of plant cells that had long since died, but the name remained, and it would eventually become one of the most important concepts in biology. Around the same time, a Dutch tradesman named Antony van Leeuwenhoek began constructing microscopes of extraordinary quality for the period. Leeuwenhoek had no formal scientific training, yet his skill in grinding lenses allowed him to see microscopic life in remarkable detail. When he examined drops of pond water, scrapings from teeth, and other tiny samples, he observed living organisms moving through the microscopic world. He called them animalcules, meaning little animals. Leeuwenhoek had unknowingly discovered bacteria and other microorganisms, revealing that an entire invisible world existed beyond the limits of ordinary vision. These discoveries slowly began reshaping the understanding of life. Over the following centuries, improvements in microscopy allowed scientists to observe cells in plants and animals with increasing clarity. By the early 19th century, German botanist Matthias Schleiden and zoologist Theodor Schwann began drawing a powerful conclusion. All living organisms appeared to be composed of cells. Plants were made of cells. Animals were made of cells. Even the simplest microscopic organisms consisted of a single cell. This idea became known as cell theory, one of the foundational principles of biology. Cell theory states that all living organisms are composed of one or more cells, and that the cell is the basic unit of life. Later work by scientist Rudolf Virchow expanded this idea further by proposing that every cell arises from a pre-existing cell. Life, in other words, continues through the division of cells. But this raised an important question. If new cells are created from existing cells, how do they inherit the characteristics of the organism they belong to? Why do human cells produce humans rather than oak trees? Why do oak tree cells produce oak trees rather than jellyfish? Somewhere inside each cell there must be information guiding development. As microscopes improved throughout the 19th century, scientists began noticing something interesting inside many cells. Near the center of the cell sat a distinct structure known as the nucleus. The nucleus appeared to play an important role in cell division. When cells prepared to divide, structures inside the nucleus became visible as thread-like bodies later called chromosomes. Scientists began suspecting that these structures might carry hereditary information. By the late 19th century, this suspicion was growing stronger. Researchers observed that chromosomes were passed from parent cells to daughter cells during division. They also noticed that reproductive cells contained special chromosome sets that combined during fertilization. Something about these structures clearly played a role in inheritance. At the same time, experiments on heredity were quietly unfolding in an unexpected place. In the 1800s, an Augustinian monk named Gregor Mendel conducted careful breeding experiments using pea plants in the garden of his monastery. Mendel tracked traits such as flower color and seed shape across multiple generations of plants. Through careful observation and mathematical analysis, he discovered consistent patterns of inheritance. Traits appeared to be passed from parents to offspring in predictable ratios. Although Mendel did not know anything about DNA or chromosomes, his work revealed that inheritance followed precise rules. Those rules hinted at the existence of discrete units of biological information, which would later become known as genes. At the year's time, however, Mendel's work remained largely unnoticed. It would not be fully appreciated until decades later, when scientists began connecting Mendel's inheritance patterns to the behavior of chromosomes during cell division. Slowly, a picture began to emerge. Cells contained structures that carried hereditary information. That information was copied and passed along as cells divided. And somehow, those instructions guided the development of living organisms. But the exact nature of that information remained a mystery. What substance inside the cell carried these instructions? What kind of molecule could store information complex enough to guide the construction of an organism? And how could something so small coordinate processes involving trillions of cells across decades of life? These questions lingered through the early years of modern biology. At that time, many scientists suspected that proteins were responsible for heredity. Proteins were complex molecules capable of forming intricate shapes and performing many tasks inside cells. DNA, by contrast, seemed far too simple. It appeared to contain only 4 repeating chemical components, arranged in long chains. How could such a simple molecule possibly encode the complexity of life? Yet slowly, evidence began pointing toward DNA as the true carrier of hereditary information. And when scientists finally uncovered the structure of the molecule in the middle of the 20th century, the answer to one mystery opened the door to many more. Because once DNA's structure became clear, it revealed something remarkable. The molecule could store information, not in words or sentences, of course, but in sequences of chemical units arranged along a long molecular chain. Like letters forming words in a language, the arrangement of those chemical units could encode instructions. Instructions that cells could read. Instructions that proteins could carry out. Instructions that ultimately guide the formation of tissues, organs, and entire living organisms. And yet the deeper scientists looked, the stranger the question became. DNA does not understand the instructions it carries. It does not know what a bone is, or what a heart does, or how eyes should form. It is simply a molecule. And yet from this molecule emerges one of the most complex structures known in the universe, a living human being. So the mystery remains quietly waiting at the center of biology. How can chemistry, following nothing more than physical laws, give rise to the astonishing order and organization of life? And how can a molecule written in only 4 chemical letters guide the construction of trillions of cooperating cells? The answer lies in a story that unfolds across genetics, molecular biology, development, and evolution. A story that begins with the simple molecule, but ultimately reaches into the deepest mechanisms that shape life itself. Long before anyone had seen DNA, before microscopes revealed the structures, inside cells with any clarity, scientists were already beginning to notice a strange regularity in the way traits pass from one generation to the next. Parents resemble their children. Oak trees produce oak trees. Cats do not unexpectedly give birth to rabbits. These patterns seem obvious today, but for most of human history the mechanism behind inheritance remained deeply mysterious. In the middle of the 19th century, an Augustinian monk named Gregor Mendel began investigating this mystery in a quiet monastery garden in what is now the Czech Republic. Mendel was not searching for DNA, and he certainly did not know anything about genes or chromosomes. Instead, he was simply trying to understand why certain characteristics appear in predictable ways across generations of plants. Pea plants turned out to be ideal for this kind of investigation. They grow quickly, produce many offspring, and display clear visible traits. Some pea plants produce smooth seeds, while others produce wrinkled ones. Some flowers appear purple, while others are white. The plants are also capable of both self-pollination and cross-pollination, allowing traits to be carefully tracked through multiple generations. Mendel spent years carefully breeding these plants and counting the traits that appeared in their offspring. His notebooks filled with observations, numbers, and patterns. What he discovered was remarkable. Traits did not blend together the way many scientists expected. Instead, they behaved as if they were controlled by discrete units of inheritance. When Mendel crossed plants with smooth seeds and wrinkled seeds, the first generation of offspring all displayed smooth seeds. Yet when those offspring produced a second generation, the wrinkled trait reappeared in a consistent proportion. The pattern repeated itself across many different traits. Mendel eventually realized that hereditary information behaved as if it were carried in pairs of invisible units. Each parent contributed one unit to the offspring, and these units separated again when the next generation formed. Today these units are known as genes. At the time, however, Mendel had no idea what genes actually were. He simply knew that something inside organisms must carry these instructions. Mendel published his findings in the 1860s, but almost no one noticed. For several decades, his work sat quietly in scientific journals, largely ignored. Biology at the time was still struggling to understand even the basic structure of cells, and the significance of Mendel's mathematical patterns was not immediately recognized. Then, around the turn of the 20th century, 3 botanists working independently began rediscovering the same principles. Hugo de Vries in the Netherlands, Karl Correns in Germany, and Erich von Tschermak in Austria each performed breeding experiments that produced the same inheritance patterns Mendel had described decades earlier. As they searched through earlier scientific literature, they discovered Mendel's original work. Suddenly, the principles of inheritance began attracting serious attention. At the same time, another important discovery was unfolding inside the microscopic world of cells. Biologists studying cell division noticed that certain structures inside the nucleus behaved in a very specific way. These thread-like structures, which had become known as chromosomes, duplicated themselves before cell division and then separated into daughter cells. Even more interestingly, reproductive cells contained half the usual number of chromosomes, and fertilization restored the full set when egg and sperm combined. This pattern strongly suggested that chromosomes might carry the hereditary units Mendel had described. One of the scientists who explored this idea in detail was Thomas Hunt Morgan at Columbia University in the United States. Morgan and his research group studied fruit flies, a tiny insect species called Drosophila, Melanogaster that breeds rapidly and produces many offspring. Fruit flies proved to be excellent experimental organisms. By tracking traits such as eye color across generations of flies, Morgan demonstrated that certain inherited characteristics were associated with specific chromosomes. This discovery linked Mendel's abstract inheritance patterns to physical structures inside the cell. Genes, it seemed, were located on chromosomes. But an even deeper question remained. What were chromosomes made of? At the time, scientists knew that chromosomes contained two main types of molecules. One was protein, a complex class of molecules capable of forming intricate structures. The other was a chemical substance called deoxyribonucleic acid. Most researchers assumed proteins carried hereditary information. Proteins were chemically diverse and capable of performing many functions inside cells. DNA, by contrast, seemed far too simple. It appeared to consist of repeating units built from only 4 chemical components. It did not look like the kind of molecule capable of storing vast biological instructions. Then, in 1928, a British bacteriologist named Frederick Griffith conducted a curious experiment involving pneumonia-causing bacteria. Griffith was studying 2 forms of the bacterium Streptococcus pneumonia. One form produced a smooth outer coating and could cause disease in mice. The other form lacked this coating and was harmless. When Griffith injected mice with the harmless bacteria, the mice survived. When he injected them with the disease-causing bacteria, the mice became ill. But something strange happened when Griffith killed the disease-causing bacteria with heat and mixed them with the harmless bacteria before injecting them into mice. The mice still became sick. Even more surprising, living disease-causing bacteria could later be recovered from the infected animals. Somehow, something from the dead bacteria had transformed the harmless bacteria into the dangerous form. Griffith called this mysterious process transformation, but he did not know what substance was responsible. The answer began emerging more than a decade later through careful biochemical experiments conducted by Oswald Avery, Colin MacLeod, and Macklin McCarty at the Rockefeller Institute in New York. Their work involved isolating different components of bacterial cells and testing which component could cause transformation. They systematically removed proteins, lipids, carbohydrates, and other cellular substances. Each time they removed one of these molecules, the transformation still occurred. But when they destroyed DNA, the transformation stopped. The conclusion was difficult to ignore. DNA appeared to carry the hereditary instructions responsible for the transformation. Many scientists remained skeptical at first. DNA still seemed too simple to hold complex biological information. Then, in 1952, another elegant experiment provided powerful confirmation. 2 researchers named Alfred Hershey and Martha Chase were studying bacteriophages, viruses that infect bacteria. These viruses are extremely simple structures composed primarily of DNA and protein. Hershey and Chase designed an experiment that allowed them to track which molecule actually entered bacterial cells during infection. They labelled viral DNA with radioactive phosphorus and viral proteins with radioactive sulfur. When the viruses infected bacteria, they could track which radioactive signal appeared inside the cells. The result was striking. The radioactive phosphorus associated with DNA entered the bacterial cells, while the radioactive sulfur associated with protein remained outside. This meant that DNA, not protein, carried the genetic instructions needed for the virus to reproduce. By the early 1950s, the evidence had become overwhelming. DNA was the genetic molecule. The instructions for building organisms were written not in proteins, but in this long chain of repeating chemical units that scientists had once considered too simple to matter very much. And now the next question began to emerge. If DNA truly carries the instructions for life, how exactly does it store that information? The answer would soon arrive in the form of one of the most famous discoveries in the history of science. By the early 1950s, scientists had gathered strong evidence that deoxyribonucleic acid carried the hereditary instructions of living organisms. Yet one crucial piece of the puzzle remained missing. No one knew what DNA actually looked like. Understanding the structure of a molecule is often the key to understanding what that molecule does. In chemistry and biology, the arrangement of atoms determines how molecules interact, how they behave, and how they carry out their roles in living systems. So if DNA truly held the instructions for life, its structure might reveal how those instructions were stored and copied. Several research groups across Europe and the United States began trying to solve this molecular mystery. Among them were two young scientists working at the Cavendish Laboratory at the University of Cambridge in England. Their names were James Watson, an American biologist with a sharp curiosity for genetics, and Francis Crick, a British physicist who had recently shifted his focus toward biology. Watson and Crick approached the problem in a slightly unusual way. Rather than performing complex laboratory experiments themselves, they relied heavily on building physical models. Using pieces of metal, cardboard, and wire, they attempted to construct three-dimensional representations of possible DNA structures. Their approach was similar to solving a puzzle by arranging pieces until they fit together in a way that made chemical sense. But to build accurate models, they needed reliable experimental data. Some of the most important clues were being produced in London at King's College by a brilliant scientist named Rosalind Franklin. Franklin was an expert in X-ray crystallography, a technique used to study the structure of molecules by observing how X-rays scatter when they pass through a crystallized sample. When X-rays interact with a structured molecule, they produce patterns of scattered light that reveal the arrangement of atoms inside the molecule. Working with her graduate student Raymond Gosling, Franklin prepared carefully hydrated DNA samples and exposed them to X-rays. The resulting diffraction patterns contained remarkable detail about the shape of the molecule. One particular image became famous in the history of science. It showed a distinct pattern of X-shaped diffraction spots that strongly suggested a helical structure. A helix is a spiral shape, similar to a twisted staircase. Franklin's careful measurements allowed her to calculate several critical dimensions of the DNA molecule. She determined the spacing between repeating units and the overall diameter of the helix. Meanwhile, another scientist at King's College named Maurice Wilkins was also studying DNA, using X-ray diffraction techniques. Wilkins had previously begun investigating DNA fibers and contributed important experimental observations that helped guide structural interpretations. The combined efforts of these researchers gradually reveal the constraints any accurate DNA model would need to satisfy. Back in Cambridge, Watson and Crick used this information to refine their model-building approach. They examined the known chemical components of DNA and attempted to arrange them in a way that matched the experimental data. DNA is built from repeating units called nucleotides. Each nucleotide contains a sugar molecule, a phosphate group, and one of 4 nitrogen-containing bases. These bases are called adenine, thymine, cytosine, and guanine. At first, scientists believed the bases might pair randomly along the molecule, but another key insight came from a chemist named Erwin Chargaff. Chargaff had carefully measured the chemical composition of DNA from many different organisms. His analyses revealed a striking pattern. In every DNA sample he examined, the amount of adenine was almost exactly equal to the amount of thymine. Likewise, the amount of cytosine closely matched the amount of guanine. This relationship became known as Chargaff's rule. Watson realized that these patterns might reflect specific pairing relationships between the bases. Adenine could pair with thymine, and cytosine could pair with guanine. When Watson and Crick incorporated these pairings into their model, something remarkable happened. The structure suddenly made sense. 2 long strands of nucleotides could wind around one another to form a double helix. The sugar and phosphate components formed the outer backbone of the molecule, while the paired bases connected the 2 strands in the center like rungs of a ladder. Adenine paired neatly with thymine, and cytosine paired with guanine. Because each base pair had similar dimensions, the helix maintained a consistent width along its entire length. The elegance of the structure was immediately obvious. More importantly, the geometry of the molecule revealed something profound about how genetic information might be copied. If the 2 strands of DNA were separated, each strand could serve as a template for constructing its partner. Wherever one strand contained adenine, the matching strand would incorporate thymine. Wherever cytosine appeared, guanine would be added. In this way, the molecule itself contained the instructions for producing an exact copy. Francis Crick reportedly recognized the significance of this insight almost immediately. According to later recollections, he excitedly remarked that they had discovered the secret of life. In 1953, Watson and Crick published their model of the DNA double helix in the scientific journal Nature. The paper was surprisingly brief, just a single page in length, yet its impact was enormous. Their article ended with a modest but memorable sentence. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. That quiet observation hinted at a revolutionary idea. Genes might be replicated through the physical properties of the DNA molecule itself. Soon after, further experiments confirmed that DNA replication operates through this template mechanism, in which each strand guides the formation of its complementary partner. The discovery of the double helix transformed biology. For the first time, scientists could see how hereditary information might be stored, copied, and transmitted across generations. The implications reached far beyond genetics. DNA's structure revealed that life operates through molecular systems governed by chemistry and physics. Biological processes could now be studied at the level of atoms and molecules, opening the door to modern molecular biology. In the decades that followed, researchers would learn how DNA sequences encode proteins, how genes are regulated, and how genetic information evolves over time. But it all began with that elegant spiral molecule, twisting through the nucleus of every living cell. A structure simple enough to describe in a few sentences, yet powerful enough to guide the unfolding of life itself. Once the structure of DNA had been revealed, a new and rather fascinating realization began to unfold. The molecule was not merely a physical object inside the nucleus of a cell, it was also something else entirely. It was a storage system for information. This idea may sound obvious today, but at the time it represented a profound shift in how scientists thought about life. Biology was no longer only about tissues, organs, and visible anatomy. It was also about coded instructions written in molecular form. To understand this idea clearly, it helps to think about the basic components of DNA. Each strand of DNA is built from repeating chemical units called nucleotides. Every nucleotide contains 3 parts: a sugar molecule, a phosphate group, and one of 4 nitrogen-containing bases. Those bases are called adenine, thymine, cytosine, and guanine. Although the chemical structure of each base is different, their real importance lies not in their shape alone, but in their order along the DNA molecule. DNA functions rather like a long molecular sentence written using 4 letters. A sequence of bases might read adenine, cytosine, guanine, thymine, adenine, and so on, continuing for enormous distances along the molecule. The particular order of those bases determines the information stored within that stretch of DNA. The human genome contains roughly 3 billion of these base pairs. That is an immense amount of information packed into a structure so thin that thousands of DNA molecules could fit across the width of a human hair. What makes this sequence so powerful is that cells can read it. Certain molecular machines inside the cell move along the DNA strand, identifying specific patterns of bases and interpreting them as instructions. In other words, the DNA sequence acts as a biological language. Long before the structure of DNA was solved, however, a physicist had already predicted that life must depend on some form of molecular information storage. In 1944, the Austrian physicist Erwin Schrödinger published a short but influential book called What is Life? Schrödinger had spent years studying the strange laws of quantum mechanics, but he became fascinated by a different question. How can living organisms preserve order and complexity generation after generation without gradually dissolving into randomness? He reasoned that heredity must depend on some kind of stable molecular structure capable of storing detailed instructions. Schrödinger proposed that the hereditary material might exist in the form of what he called an aperiodic crystal. Ordinary crystals, such as salt or quartz, are built from repeating patterns of atoms arranged in identical units. An aperiodic crystal, by contrast, would contain a stable structure whose internal pattern could vary from place to place. That variation could store information. Although Schrödinger did not know about DNA's structure at the time, his idea turned out to be remarkably accurate. DNA is indeed a stable molecular structure whose sequence can vary almost endlessly along its length. Each variation in that sequence carries different biological instructions. Around the same time that molecular biology was beginning to emerge, another scientific field was also exploring the nature of information itself. In the late 1940s, an American mathematician named Claude Shannon developed a mathematical framework known as information theory. Shannon was studying how messages could be transmitted efficiently through communication systems such as telephones and radio signals. His work introduced the idea that information can be measured, encoded, and transmitted using symbolic sequences. Although Shannon's work focused on electronic communication, the parallels with genetics soon became clear. DNA sequences behave in many ways like encoded messages, storing information that must be copied and transmitted accurately during cell division. This similarity led scientists to begin thinking about life in informational terms. Genes, in this framework, can be understood as specific segments of DNA that contain instructions for building functional molecules. Each gene occupies a particular region along the DNA molecule and carries the information required to produce a specific product inside the cell. In most cases, that product is a protein. Proteins are large molecules made from chains of amino acids folded into intricate three-dimensional shapes. These shapes allow proteins to perform a vast range of tasks, from accelerating chemical reactions to forming structural components of tissues. The sequence of bases in a gene determines the sequence of amino acids in a protein. In effect, the gene contains a molecular recipe for building that protein. Once scientists began recognizing this relationship, another fundamental principle of biology emerged. This principle became known as the central dogma of molecular biology. The central dogma was articulated by Francis Crick in the late 1950s as scientists were beginning to unravel the mechanisms of gene expression. It describes the flow of biological information through three major molecular stages. Information stored in DNA is first copied into a related molecule called ribonucleic acid, or RNA. The RNA sequence is then used as a template for assembling a protein. The process follows a directional flow from DNA to RNA to protein. This sequence of information transfer allows cells to translate the genetic instructions stored in DNA into the molecular machinery required for life. In practical terms, this means that the instructions encoded in DNA ultimately shape the physical structures inside the cell. Proteins produced from genetic instructions form enzymes that drive metabolism. They create structural components of tissues. They regulate gene activity, communicate signals between cells, and coordinate complex biological processes. The DNA sequence itself does not directly build bones or muscles. Instead, it guides the production of the molecules that do. Seen from this perspective, DNA can be understood as an extraordinary storage system for biological instructions. Its sequence acts as a molecular script that cells read and interpret through a series of chemical processes. And yet, despite its role as an information system, DNA remains nothing more than a molecule composed of atoms bonded together according to the laws of chemistry. It does not understand the information it carries. It does not plan or anticipate the structures that will eventually emerge. Instead, the instructions embedded in its sequence interact with cellular machinery that has itself evolved over immense spans of time. From these interactions, something remarkable happens. Proteins form, cells specialize, tissues organize, and gradually, through countless coordinated processes, the instructions written in that long molecular sequence begin to unfold into the astonishing complexity of living organisms. If DNA truly stores the instructions for life, those instructions must eventually be read. After all, a book full of recipes is only useful if someone actually opens the pages and begins cooking. In a similar way, the information written along the DNA molecule must be interpreted by the cell before it can influence anything in the physical world. This process of reading genetic information is known as gene expression. It is the mechanism by which the sequence of bases in DNA becomes a working molecule inside the cell. The first stage of this process is called transcription. Inside the nucleus of the cell, long strands of DNA remain tightly coiled and carefully protected. Certain sections of the DNA contain genes, each representing a specific stretch of sequence that carries instructions for producing a particular molecule. When the cell needs to use one of these instructions, it does not remove the original DNA from the nucleus like a library book taken off a shelf. Instead, the cell makes a temporary copy of the information. This copying process is carried out by an enzyme known as RNA polymerase. RNA polymerase moves along the DNA strand, reading the sequence of bases one unit at a time. As it travels, it assembles a complementary strand of a related molecule called ribonucleic acid, more commonly known as RNA. The newly produced RNA molecule contains a sequence of bases that corresponds to the information stored in the gene. This RNA copy is called messenger RNA. Messenger RNA acts exactly as the name suggests. It carries genetic instructions from the DNA in the nucleus to other parts of the cell where those instructions can be used. The discovery of messenger RNA was an important milestone in molecular biology. During the early 1960s, two French scientists, François Jacob and Jacques Monod, were studying gene regulation in bacteria at the Pasteur Institute in Paris. Their experiments revealed that cells produce temporary RNA copies of genes when those genes are needed. These messenger molecules travel through the cell and guide the production of proteins. This insight helped clarify how genetic information moves from the relatively stable DNA molecule into the dynamic machinery of the cell. Once messenger RNA has been produced, the next stage begins. This stage is known as translation. The name translation is appropriate because the information stored in one molecular language must now be converted into another. DNA and RNA use sequences of nucleotide bases, while proteins are built from entirely different building blocks called amino acids. Translation therefore requires a system capable of converting sequences of bases into sequences of amino acids. This remarkable process occurs within structures known as ribosomes. Ribosomes are tiny molecular machines found in every living cell. Each ribosome is composed of ribosomal RNA and specialized proteins that work together to assemble new protein molecules. When messenger RNA arrives at a ribosome, the ribosome attaches to the RNA strand and begins moving along it. As the ribosome travels along the messenger RNA, it reads the sequence of bases in groups of 3. Each group of 3 bases forms what scientists call a codon. A codon represents a specific instruction telling the ribosome which amino acid should be added next to the growing protein chain. The relationship between codons and amino acids forms what is known as the genetic code. During the early 1960s, scientists worked intensely to decipher this code. One of the pioneers in this effort was Marshall Nierenberg at the National Institutes of Health in the United States. Nierenberg conducted experiments using artificial RNA molecules placed inside cell extract containing ribosomes and other cellular components. By observing which amino acids were assembled in response to specific RNA sequences, his team began identifying which codons corresponded to particular amino acids. At roughly the same time, Hargobin Korana used chemical synthesis techniques to create precise RNA sequences that helped confirm many codon assignments. Another important contribution came from Robert Holley, who studied the structure of transfer RNA molecules that deliver amino acids to the ribosome during protein assembly. Together, the work of Nierenberg, Korana, and Holley revealed the full structure of the genetic code. The code contains 64 possible codons created from combinations of the 4 RNA bases. These codons correspond to the 20 amino acids used to build proteins, along with special signals that mark the beginning and end of protein synthesis. One codon might instruct the ribosome to add the amino acid leucine. Another might specify glycine or serine. Still others signal the ribosome to stop translation once the protein chain is complete. As the ribosome moves steadily along the messenger RNA strand, transfer RNA molecules deliver the correct amino acids one by one. Each new amino acid is joined to the growing chain through chemical bonds, gradually forming a protein. When the ribosome reaches a stop codon, the completed protein is released. At that point, the newly formed protein begins folding into its final three-dimensional shape. That shape determines how the protein will function inside the cell. Some proteins become enzymes that accelerate chemical reactions. Others form structural frameworks within cells and tissues. Some act as molecular sensors detecting signals from the environment, while others regulate gene activity itself. In fact, proteins perform the majority of tasks required for life. They digest food molecules, repair damaged DNA, transmit signals across cell membranes, and help construct the structural components of tissues and organs. In this way, the instructions encoded in DNA ultimately shape the physical reality of the cell. A gene is written in the language of nucleotide bases. That sequence is copied into messenger RNA during transcription. The RNA message is then translated by ribosomes into a chain of amino acids, and that chain folds into a protein capable of performing a specific task within the living system. What began as a sequence of chemical letters inside the nucleus becomes a working molecular machine inside the cell. Seen from this perspective, the instructions in DNA do not directly build living structures. Instead, they guide the production of proteins that do the work. And through the coordinated activity of thousands of different proteins, the cell carries out the countless processes required to sustain life. The sequence of bases in DNA may look simple on paper, but through transcription and translation it becomes the foundation of an extraordinarily complex molecular world quietly unfolding inside every living cell. Once a gene has been translated into a chain of amino acids, the result is a protein. At first glance, this chain might appear rather simple, a sequence of chemical building blocks linked together in a long molecular thread. But what happens next is where the true complexity begins to emerge. The chain does not remain stretched out like a piece of string. Instead, it begins to fold. Sections of the chain twist, bend, and curl into loops and spirals. Certain amino acids attract one another, while others repel. Chemical interactions guide the chain into a precise three-dimensional shape. Within seconds, what began as a linear sequence transforms into a complex molecular structure with ridges, pockets, grooves, and surfaces that allow it to interact with other molecules. This folding process is essential because the shape of a protein determines what it can do. A useful way to imagine this is to think of a key fitting into a lock. Only a key with the correct shape can open a particular lock. In a similar way, proteins often function by binding to specific molecules whose shapes match their own structures. This principle lies at the heart of much of cellular chemistry. One of the most important roles proteins play is acting as enzymes. Enzymes are biological catalysts, meaning they accelerate chemical reactions that would otherwise occur extremely slowly. Inside the body, thousands of chemical reactions are constantly taking place. Nutrients must be broken down. Energy must be released from molecules. New molecules must be assembled to build cellular structures. Waste products must be processed and removed. Many of these reactions would take years or even centuries to occur at noticeable rates without assistance. Enzymes solve this problem by lowering the energy barrier required for reactions to proceed. An enzyme binds to specific molecules known as substrates. The enzyme's shape positions the substrates in a way that makes the chemical reaction easier to occur. Once the reaction is complete, the enzyme releases the products and becomes ready to catalyze another reaction. Some enzymes can perform this task thousands of times each second. Without enzymes, metabolism would grind to a halt. Cells would simply not be able to maintain the chemical activity required for life. Proteins also provide structural support for tissues throughout the body. One example is collagen, the most abundant protein in the human body. Collagen fibers form long, strong strands that help give connective tissues their strength and flexibility. These fibers contribute to the structure of skin, tendons, cartilage, and bones. Another important structural protein is keratin. Keratin forms the durable material that makes up hair, nails, and the outer layer of skin. Its tightly packed molecular arrangement allows it to resist wear and protect underlying tissues. While enzymes and structural proteins are essential, many proteins also serve as regulators inside the cell. These regulatory proteins help control when genes are turned on or off, ensuring that the correct proteins are produced at the right time and in the right place. Some regulatory proteins act as transcription factors, binding to specific regions of DNA and influencing whether nearby genes are copied into RNA. Others participate in signaling pathways that allow cells to communicate with one another. When a cell receives a signal from its environment, perhaps from a neighboring cell or a circulating hormone, regulatory proteins help interpret that signal and adjust gene activity accordingly. Through these interactions, proteins help coordinate the behavior of entire tissues. All of these functions depend on the precise folding of protein molecules. If a protein fails to fold correctly, it may lose its ability to function. In some cases, misfolded proteins can accumulate inside cells and interfere with normal processes. Several human diseases, including certain neurodegenerative disorders, are linked to problems involving protein folding. Understanding how proteins achieve their correct shapes became an important scientific question during the 20th century. One of the scientists who made major contributions to this field was Christian Anfinsen, an American biochemist working at the National Institutes of Health. Anfinsen conducted experiments in the 1960s using an enzyme called ribonuclease. He carefully unfolded the protein using chemical treatments that disrupted its structure. Once the unfolding chemicals were removed, the protein spontaneously refolded back into its original shape and regained its enzymatic activity. This experiment demonstrated something remarkable. The instructions for a protein's three-dimensional structure are encoded directly within its amino acid sequence. In other words, the linear chain of amino acids produced during translation already contains all the information needed for the protein to fold correctly. This discovery became known as Anfinsen's dogma. It revealed that the genetic instructions stored in DNA ultimately determine not only the sequence of amino acids in a protein, but also the three-dimensional shape that protein will adopt, and that shape, in turn, determines what the protein can do inside the cell. Seen from this perspective, DNA functions less like a blueprint for building tissues directly and more like a library of recipes. Each gene contains instructions for producing a particular protein. Those proteins then carry out the physical work required to maintain the cell and build the structures of the organism. Enzymes accelerate metabolic reactions. Structural proteins reinforce tissues. Regulatory proteins coordinate gene activity and cellular communication. Together, these molecules form an intricate network of interactions that sustain life. DNA itself does not assemble bones or pump blood or transmit nerve signals. Instead, it directs the production of the molecular machines that perform those tasks. Within every cell, thousands of different proteins operate simultaneously, interacting with one another in carefully regulated systems. From these molecular activities emerge the larger structures and functions that define living organisms. The instructions may be written in DNA, but the real builders of the cell are proteins. working quietly and continuously in the microscopic world beneath our awareness. One of the more curious facts about the human body is that almost every cell contains the same DNA. A neuron deep within the brain carries essentially the same genetic instructions as a muscle cell in the leg, a skin cell on the fingertip, or a cell lining the inside of the stomach. In each of these cases, the full human genome is present, a molecular archive containing roughly 3 billion base pairs of information. And yet these cells behave very differently. A neuron develops long branching extensions called dendrites and axons that allow it to transmit electrical signals across vast networks in the nervous system. A muscle cell becomes elongated and specialized for contraction, allowing tissues to generate force and movement. Epithelial cells form protective layers that line surfaces throughout the body, from the skin to the interior of the lungs and digestive tract. Despite sharing the same DNA, these cells adopt entirely different identities. The explanation for this apparent contradiction lies in a principle known as differential gene expression. Differential gene expression refers to the idea that not all genes are active in every cell at every moment. Instead, each cell type activates a specific subset of genes while leaving many others inactive. A neuron, for example, expresses genes that produce proteins involved in transmitting nerve impulses. Muscle cells express genes that build contractile proteins such as actin and myosin. Epithelial cells activate genes that support barrier functions and cellular adhesion. The underlying DNA sequence remains the same, but the pattern of gene activity changes. This selective use of genetic information allows cells to specialize. In a sense, the genome functions like an enormous library containing thousands of books. Every cell possesses the entire library, but each cell type reads only the volumes relevant to its role. A neuron consults one set of instructions. A liver cell consults another. A skin cell reads yet another collection of genetic recipes. The question then becomes how cells decide which genes to read and which to ignore. Scientists began exploring this question during the middle of the 20th century when the field of gene regulation started to take shape. One of the most influential discoveries came from the work of François Jacob and Jacques Monod at the Pasteur Institute in Paris. Jacob and Monod were studying bacteria, specifically the bacterium Escherichia coli, which often lives in the human digestive system. Their research focused on how bacteria regulate the production of enzymes that digest different sugars. They discovered that certain genes are turned on only when needed. When the sugar lactose is present in the environment, bacteria activate genes that produce enzymes capable of breaking down lactose. When lactose is absent, those genes remain silent. This elegant regulatory system became known as the lac operon. The lac operon demonstrated that genes could be controlled by regulatory molecules that respond to environmental signals. Certain proteins act as molecular switches, binding to DNA and either blocking or enabling gene transcription. These regulatory proteins detect signals within the cell or from the surrounding environment and adjust gene activity accordingly. Although Jacob and Monod were studying bacteria, the principles they uncovered turned out to be fundamental to all life. In more complex organisms such as humans, gene regulation operates through intricate networks of regulatory proteins and signaling pathways. Proteins called transcription factors bind to specific regions of DNA near genes and influence whether those genes are copied into messenger RNA. Some transcription factors activate gene expression, while others suppress it. Cells also communicate with one another through signaling molecules. Hormones, growth factors, and other chemical signals travel between cells, activating signaling pathways that ultimately influence gene activity. When a signal arrives at the surface of a cell, it may trigger a cascade of molecular events inside the cell. These events often involve chains of interacting proteins that transmit the signal inward until it reaches the nucleus, where gene expression can be adjusted. Through these signaling systems, cells respond to their environment and coordinate their activities with neighboring cells. During development, these regulatory processes become especially important. As an embryo grows from a single fertilized cell into a complex organism, different groups of cells must activate different sets of genes. Some cells begin forming the nervous system. Others develop into muscle tissue, connective tissue, or internal organs. Each decision depends on the careful control of gene expression. Signals from neighboring cells, gradients of signaling molecules, and regulatory proteins all contribute to determining which genes are active in each cell at a given moment. This process is remarkably precise. The developing organism does not change its DNA sequence in order to create different tissues. Instead, it relies on the selective activation of genes within the shared genome. Even in adulthood, gene expression continues to shift as cells respond to changing conditions. Immune cells activate certain genes when encountering pathogens. Liver cells adjust gene activity depending on metabolic needs. Skin cells alter their gene expression as they mature and move toward the outer surface of the body. The genome remains constant, but its interpretation varies. Seen from this perspective, the genome is not a rigid blueprint that dictates a fixed outcome. Instead, it is more like a dynamic instruction manual whose pages are consulted differently depending on context. Regulatory proteins, signaling pathways, and cellular environments together determine which sections of that manual are read. Through this system of differential gene expression, the same DNA sequence can give rise to the astonishing diversity of cell types that make up the human body. What began as a single fertilized cell gradually becomes a complex organism composed of trillions of specialized cells, each performing its own tasks. The instructions were present from the beginning, but it is the careful orchestration of gene expression that allows those instructions to unfold into the extraordinary variety of life. Every human life begins as a single cell. This cell forms when a sperm cell and an egg cell merge during fertilization, combining their genetic material to create a new organism with a complete set of DNA. At that moment, the fertilized egg, known as a zygote, contains the entire genome required to guide the development of a human body. Yet if one were to look at that tiny cell under a microscope, it would not appear especially remarkable. It would look like a small sphere of cytoplasm surrounded by a membrane, with a nucleus containing DNA. No bones, no organs, no visible hint of a brain, heart, or skin. And yet within that single cell lies the beginning of a process that will eventually produce trillions of specialized cells arranged into complex tissues and organs. Very soon after fertilization, the zygote begins dividing. The first division produces 2 cells. Those 2 cells divide again to produce 4. The 4 become 8. The number continues doubling in a series of rapid divisions known as cleavage. At this stage, the cells are called blastomeres. They are roughly similar in appearance and still retain the full genetic instructions inherited from the original fertilized egg. If this early cluster of cells were observed carefully, it might appear as though each division simply produces smaller and smaller copies of the original cell. But something far more subtle is already taking place. Even before the embryo begins forming recognizable structures, the earliest instructions for development are already at work. One of the most fascinating aspects of early embryonic development is that many of these instructions exist before the embryo's own genes even begin functioning fully. The fertilized egg does not begin development from a completely blank state. Instead, the egg cell already contains a rich collection of molecules provided by the mother. These molecules include messenger RNA, proteins, and signaling factors that were deposited into the egg during its formation in the ovary. Collectively, they create chemical patterns that help guide the earlier stages of development. These early molecular influences are known as maternal effects. Maternal effect genes are genes expressed in the mother that produce molecules stored in the egg before fertilization occurs. Once the embryo begins developing, these molecules help determine the earliest patterns of organization. In other words, some of the earliest developmental instructions originate not from the embryo's own newly activated genes, but from molecular information prepared by the mother. Within the fertilized egg, these molecules are not distributed evenly. Instead, they form gradients. A gradient is a gradual change in concentration across space. In the case of embryonic development, certain molecules may be present in higher concentration at one end of the egg and lower concentration at the opposite end. These gradients act as positional signals. Cells that form in different locations within the embryo experience different concentrations of these signaling molecules. By detecting these differences, cells begin to interpret their position within the developing organism. This process gradually establishes the major axes of the body. In most animals, including humans, the earliest axis distinguishes the head region from the tail region. Other axes help define the back from the belly and the left side from the right side. Although the embryo at this stage remains extremely small and visually simple, the molecular groundwork for these spatial distinctions is already being laid. One of the most influential discoveries related to these gradients came from research using fruit flies. The fruit fly species Drosophila melanogaster has been studied extensively by developmental biologists because its embryos develop quickly and its genetics are well understood. In the 1970s and 1980s, scientists such as Christian Nusslein-Volhard and Eric Wieschaus conducted large-scale genetic experiments studying fruit fly development. By introducing mutations and observing the resulting embryos, they identified many genes responsible for early pattern formation. Among the most famous examples is the bicoid gene, a maternal effect gene that produces a protein forming a concentration gradient within the fruit fly embryo. High concentrations of bicoid protein appear near what will become the head region, while lower concentrations extend toward the tail. Cells interpret this gradient to determine their position along the body axis. This discovery demonstrated that spatial patterns in embryos can arise from relatively simple chemical gradients. Similar principles appear in many other organisms as well. Zebrafish have become another widely used model organism for studying early development. Their embryos develop outside the body and remain transparent during early stages, allowing scientists to observe cellular events directly under the microscope. Researchers studying zebrafish embryos have identified signaling pathways that guide the formation of tissues and body structures. Gradients of signaling molecules help determine which cells will become part of the nervous system, which will contribute to muscle tissue, and which will form internal organs. Although humans develop inside the body and are therefore more difficult to study directly, many of the same developmental mechanisms appear to operate across animal species. This conservation across evolution is one of the most striking features of developmental biology. Genes and signaling pathways discovered in fruit flies or fish often have closely related counterparts in humans. These findings reveal that the earliest instructions for building a body emerge through chemical interactions rather than through a fully detailed blueprint. The embryo does not begin with a miniature map of organs waiting to unfold. Instead, development unfolds through dynamic processes in which cells interpret signals from their surroundings. Chemical gradients establish positional information. Regulatory genes activate different developmental programs depending on location. Cells respond to signals from neighboring cells, gradually coordinating their behavior to form organized tissues. This entire process begins long before any recognizable anatomical structures appear. During these earlier stages, the embryo remains a microscopic cluster of dividing cells. Yet within that cluster, molecules are already directing complex decisions about identity, position, and future development. It is rather like watching the opening moments of a symphony. At first, only a few quiet notes are heard. The melody has not yet taken shape, and the full orchestra has not entered. But those early notes contain the seeds of everything that will follow. In much the same way, the earliest molecular signals within the embryo begin guiding a developmental process that will eventually produce a fully formed organism. The remarkable outcome of this process depends on countless molecular interactions occurring long before any visible features appear. Before the first heartbeat, before the first nerve signals, before the earliest outlines of a body begin to form. Development begins quietly, in gradients of molecules and patterns of gene activity inside a cluster of dividing cells. From these subtle beginnings, the astonishing complexity of life gradually unfolds. As the developing embryo continues to grow, the earliest molecular gradients begin guiding a remarkable transformation. Cells that once seemed nearly identical gradually start adopting distinct identities. Some begin forming segments that will later become parts of the head. Others move toward the development of the thorax or abdomen. Still others contribute to tissues that will eventually produce muscles, organs, and nervous systems. Behind these coordinated changes lies an extraordinary set of genes that act almost like architects of the body plan. These genes are known as homeotic genes. Homeotic genes control where specific structures appear along the body axis of an organism. They help determine which regions of the embryo will develop into particular body parts. In other words, they help answer questions such as where the head should form, where the thorax belongs, and where the abdomen begins. The discovery of these genes emerged from careful studies of fruit flies during the 20th century. One of the scientists who played a central role in this discovery was Edward Lewis at the California Institute of Technology. Lewis spent many years studying genetic mutations in the fruit fly species Drosophila melanogaster, the same small insect that had already proven useful in earlier genetic research. Lewis noticed that certain mutations produced very strange results in developing flies. In some cases, a structure that normally appeared in one part of the body would appear in an entirely different location. One famous example involved flies that developed extra pairs of wings where antennae should have been. These unusual transformations revealed that certain genes control the identity of entire body segments. Lewis eventually demonstrated that groups of related genes control the arrangement of body structures along the head-to-tail axis. These genes are now known as Hox genes. Hox genes act as master regulators during development. Each gene within the Hox cluster becomes active in a specific region of the embryo. The pattern of activation tells cells which part of the body they belong to. A cell located near the future head region activates a different set of Hox genes than a cell located farther along the body axis. These gene activity patterns help establish the blueprint for the organism's body layout. The instructions provided by Hox genes do not directly build tissues themselves. Instead, they regulate many other genes that control the development of specific structures. For example, a Hox gene might activate a network of genes that guide the formation of a particular body segment. Those genes then influence the growth of tissues, the differentiation of cells, and the development of anatomical structures appropriate for that region. In this way, Hox genes operate like high-level coordinators directing large developmental programs. The importance of these genes became even clearer through further research conducted by Christian Nüsslein-Volhard and Erich Wieschaus in Germany during the 1970s and 1980s. Working at the European Molecular Biology Laboratory in Heidelberg, they performed systematic genetic experiments on fruit fly embryos. Their goal was to identify genes responsible for early pattern formation. They exposed fruit flies to chemical mutagens that produced genetic mutations, then examined the resulting embryos under the microscope. Some embryos developed unusual patterns of segments, others lacked entire body regions, still others displayed duplicated structures. By carefully analyzing these mutants, Nüsslein-Volhard and Wieschaus identified several classes of genes involved in establishing the segmented body pattern of the fruit fly. These included gap genes, pair rule genes, and segment polarity genes. Each group of genes played a specific role in organizing the developing embryo. Gap genes help establish broad regions along the body axis. Pair rule genes refine this pattern into repeating segments. Segment polarity genes then define the boundaries and orientation of each segment. Together, these genes create a hierarchical system of developmental control. Large-scale patterns are established first, followed by increasingly detailed organization as development proceeds. For their groundbreaking work in developmental genetics, Nüsslein-Volhard and Wieschaus were later awarded the Nobel Prize in Physiology or Medicine. Perhaps the most surprising discovery, however, came when scientists began comparing these developmental genes across different species. Researchers found that Hox genes are not unique to fruit flies. Very similar genes exist in many other animals, including mice, birds, fish, and humans. Even more remarkable, the order of these genes along the chromosome often mirrors the order of the body regions they control. Genes responsible for anterior structures, meaning those toward the head, tend to appear earlier in the sequence. Genes controlling posterior regions toward the tail appear later. This phenomenon is known as collinearity. The presence of similar Hox genes across diverse species suggests that the fundamental mechanisms controlling body patterning are deeply conserved throughout evolution. In other words, the genetic systems guiding development in insects share ancient evolutionary roots with those guiding development in vertebrates. Although the final body shapes of these organisms differ dramatically, the underlying genetic toolkit contains many of the same components. Evolution has modified and reused these genes over hundreds of millions of years, adjusting their activity patterns to produce different anatomical forms. A fruit fly and a human obviously do not look very similar, yet many of the genes guiding their early development belong to the same ancient family. This discovery transformed how scientists understand the relationship between genetics and evolution. Instead of inventing entirely new genes for each new organism, evolution often repurposes existing developmental systems. Small changes in when and where genes are activated can produce significant differences in body structure. A slight shift in the activity of a regulatory gene may alter the length of a limb, the arrangement of body segments, or the shape of a skeletal structure. Over many generations, these changes accumulate, giving rise to the extraordinary diversity of life seen across the planet. Through the study of homeotic genes and developmental genetics, scientists have come to understand that the body plan of an organism is not assembled randomly. It is guided by networks of regulatory genes that establish positional information across the developing embryo. These genes act as architects of biological form. They do not physically build tissues themselves, but they coordinate the activity of countless other genes that shape the organism. From the earliest stages of development, these molecular architects quietly guide cells as they take their places within the growing body. And remarkably, many of the same genetic instructions have been guiding the development of animals for hundreds of millions of years. As development continues, the embryo becomes increasingly organized. Cells begin forming tissues. Tissues begin forming organs, and organs begin integrating into functional systems. Yet beneath these visible changes lies an invisible layer of coordination taking place inside every cell. This coordination depends on a remarkable form of biological control known as gene regulation. Although every cell contains essentially the same DNA, different genes must be activated or silenced depending on the cell's role. A neuron in the brain must express genes that support electrical signaling. A muscle cell must express genes that allow contraction. A skin cell must produce structural proteins that protect the body's surface. The instructions for all of these possibilities exist within the genome, but the correct instructions must be activated at the right place and at the right moment. One of the primary tools used by cells to regulate genes involves proteins known as transcription factors. Transcription factors are specialized proteins that bind to specific regions of DNA. By attaching to these regions, they influence whether a gene becomes active or remains silent. A gene does not simply turn itself on automatically. Instead, transcription factors interact with regulatory sequences located near genes, often referred to as promoters and enhancers. These regions function as control panels where regulatory proteins gather and interact. When the correct transcription factors bind to these regulatory sequences, they recruit the molecular machinery required to begin transcription. This machinery includes the enzyme RNA polymerase, which reads the DNA sequence and produces messenger RNA. If the required transcription factors are absent, the gene remains inactive. Through this mechanism, cells can control gene activity with remarkable precision. But gene regulation rarely involves a single transcription factor acting alone. In most cases, multiple transcription factors interact together, forming complex combinations that determine whether a gene is activated or suppressed. These interactions create a sophisticated regulatory system capable of producing finely tuned patterns of gene expression. One of the scientists who devoted much of his career to understanding these regulatory systems was the developmental biologist Eric Davidson at the California Institute of Technology. Davidson introduced the concept of gene regulatory networks. A gene regulatory network is a system of interacting genes and regulatory proteins that collectively control developmental processes. Within such a network, genes influence one another's activity through chains of regulatory interactions. One gene may activate a transcription factor that controls several other genes. Those genes may then activate or suppress additional regulatory proteins, creating an intricate cascade of signals. These networks allow large groups of genes to coordinate their activity. Instead of each gene operating independently, developmental genes function as parts of interconnected systems. When one element in the network changes its activity, many downstream genes respond accordingly. Through this coordination, gene regulatory networks guide major developmental transitions. For example, during the formation of the nervous system, regulatory networks activate genes responsible for neuronal identity while suppressing genes associated with other cell types. During muscle development, a different regulatory network promotes the expression of muscle-specific proteins such as actin and myosin. The power of these networks lies in their ability to produce stable patterns of gene activity. One important mechanism contributing to this stability involves feedback loops. In a feedback loop, the product of a gene can influence its own activity or the activity of genes within the same regulatory network. Positive feedback loops reinforce gene activation. When a gene becomes active, it may produce a transcription factor that further promotes its own expression. This reinforcement helps maintain stable patterns of gene activity within a cell. Negative feedback loops perform the opposite role. They limit or suppress gene activity once certain thresholds are reached, preventing uncontrolled expression. These feedback mechanisms help ensure that developmental programs remain stable once they begin. Without such stabilization, cells might fluctuate unpredictably between different genetic states. Gene regulation also involves precise timing. During development, certain genes must activate only at particular moments. Activating a developmental gene too early or too late can disrupt the entire sequence of events required to build tissues and organs. For example, during limb development in vertebrates, specific regulatory genes must activate in a carefully controlled sequence. Early genes establish the basic structure of the limb bud. Later genes refine the formation of bones, muscles, and connective tissues. If this timing becomes disrupted, the resulting structures may develop incorrectly. Researchers studying developmental timing have discovered that gene activation often follows cascading sequences. One group of genes activates another, which then activates additional genes further down the developmental pathway. Each stage prepares the conditions required for the next. This layered progression resembles a carefully choreographed performance in which each participant enters the stage at precisely the right moment. The importance of timing becomes especially clear when examining processes such as embryonic segmentation or organ formation. In many animals, developmental genes turn on and off in rhythmic patterns, producing repeated structures such as vertebrae along the spine. These rhythmic cycles depend on oscillating gene activity within regulatory networks. Even subtle disruptions to these rhythms can alter the final body structure. What emerges from this research is a deeper understanding of how DNA helps build living organisms. The genome does not function as a simple instruction manual listing parts to assemble one after another. Instead, it contains a dynamic system of interacting regulatory elements that coordinate the activity of thousands of genes across space and time. Transcription factors interpret the DNA sequence. Gene regulatory networks integrate signals from multiple genes. Feedback loops stabilize developmental programs. Timing mechanisms ensure that each stage of development unfolds in the correct sequence. Together, these systems transform genetic information into organized biological structure. Inside the developing embryo, millions of cells are constantly reading and interpreting their genetic instructions. Yet those instructions are not followed blindly. Cells evaluate signals from their environment, interpret regulatory cues, and respond according to complex genetic networks. Through this interplay of regulation, timing, and feedback, the genome orchestrates one of the most intricate processes in nature. The gradual construction of a living body from a single fertilized cell. As development continues, cells gradually adopt stable identities. Some become neurons capable of transmitting electrical signals across long distances. Others become muscle cells designed to contract and generate force. Still others become liver cells performing complex metabolic chemistry. What makes this remarkable is that nearly every one of these cells contains exactly the same DNA. The genetic sequence inside a neuron is essentially identical to the genetic sequence inside a skin cell or a blood cell, yet their behavior, structure, and function are dramatically different. This difference arises because different genes are active in different cells. But once a cell commits to a particular identity, something important must happen. That identity must be remembered. A neuron must remain a neuron when it divides. A skin cell must remain a skin cell generation after generation. This stability depends on a system known as epigenetics. Epigenetics refers to chemical modifications that influence gene activity without changing the underlying DNA sequence itself. The word literally means above genetics, referring to regulatory layers that sit on top of the genetic code. These modifications act like molecular annotations written onto the genome. They do not alter the letters of the DNA sequence, but they influence how those letters are interpreted by the cell. One of the most widely studied epigenetic mechanisms involves DNA methylation. DNA methylation occurs when small chemical groups known as methyl groups attach to specific bases within the DNA molecule. These attachments commonly occur at regions where cytosine bases appear next to guanine A bases in the sequence. When methyl groups accumulate in certain regions of DNA, they tend to reduce the activity of nearby genes. The methyl groups can interfere with the ability of transcription factors and other regulatory proteins to bind to DNA. As a result, heavily methylated genes often remain inactive. Another important epigenetic system involves proteins known as histones. Inside the nucleus, DNA does not float freely. Instead, it wraps around clusters of histone proteins, forming a structure called chromatin. This packaging allows the long DNA molecules to fit inside the tiny space of the nucleus. Histones also play a role in regulating gene accessibility. Chemical modifications to histone proteins can influence how tightly DNA is wrapped around them. When DNA is tightly coiled, the genes within that region become difficult for transcription machinery to access. When the chromatin structure loosens, genes become more accessible and more likely to be expressed. These chemical changes include processes such as histone acetylation, histone methylation, and other modifications affecting the histone tails that extend from the chromatin structure. Through these modifications, cells can control which regions of DNA remain open for transcription and which remain closed. Together, DNA methylation and histone modification form part of the epigenetic landscape that governs gene activity. One of the scientists who made major contributions to understanding DNA methylation was Adrian Bird at the University of Edinburgh. Bird studied regions of the genome known as CPG islands, areas where cytosine and guanine bases appear frequently together. These regions often occur near gene promoters and are typically associated with genes that play essential roles in cellular function. Bird and his colleagues discovered that methylation patterns within these regions influence whether genes remain active or silent. His research helped reveal how epigenetic modifications regulate gene expression across the genome. What makes epigenetics especially fascinating is its role in cellular memory. When a cell divides, it must copy not only its DNA sequence, but also its epigenetic state. Enzymes, responsible for DNA methylation, can recognize existing methylation patterns and replicate them on newly synthesized DNA strands during cell division. Similarly, histone modifications can be re-established as chromatin is rebuilt in daughter cells. This process allows cells to preserve their identity across many generations of cell division. A muscle cell continues producing muscle proteins. a liver cell continues performing metabolic chemistry. A neuron maintains its specialized structure and electrical activity. In this way, epigenetic mechanisms help maintain stable patterns of gene expression long after the original developmental signals have passed. Yet epigenetic patterns are not always completely fixed. In some cases, environmental influences can affect epigenetic states. Nutrition, stress, exposure to toxins, and other environmental conditions may influence patterns of DNA methylation or histone modification. These changes can alter gene activity in ways that affect cellular function. Scientists studying epigenetics have explored how early life experiences may shape gene regulation later in life. Some experiments in animal models have suggested that maternal care, diet, and environmental conditions can influence epigenetic marks affecting behavior or metabolism. Although the details of these processes remain an active area of research, the findings suggest that gene expression may be shaped not only by genetic inheritance, but also by life experiences. It is important to understand that epigenetic changes do not rewrite the DNA sequence itself. The genetic code remains intact. Instead, epigenetic mechanisms influence how that code is read and interpreted by the cell. One might imagine the genome as a vast library containing thousands of books. The DNA sequence represents the words printed on the pages. Epigenetic modifications act more like bookmarks, highlights, and notes indicating which passages should be read and which should remain closed. Different cells open different parts of the library depending on their role. A neuron consults certain volumes. A muscle cell opens others. A liver cell reads yet another set of instructions. Through this selective access, cells produce the molecules needed for their specialized functions. Understanding epigenetics has transformed how scientists think about gene regulation and development. It reveals that the genome is not simply a static collection of instructions. Instead, it exists within a dynamic regulatory environment shaped by chemical modifications, regulatory proteins, and cellular history. The DNA sequence provides the fundamental blueprint, but the epigenetic state determines how that blueprint is interpreted within each cell. Together, genetic information and epigenetic regulation create the stable yet flexible systems that allow living organisms to grow, develop, and adapt across the course of a lifetime. As the embryo continues developing, cells do far more than simply read their own genetic instructions. They also listen carefully to the signals coming from their neighbors. Every developing organism depends on constant communication between cells. Without this communication, the complex coordination required to build tissues and organs would quickly fall apart. Cells communicate using signaling molecules. These molecules are released by one cell and detected by receptors on nearby cells. When a signaling molecule binds to its receptor, it triggers biochemical reactions inside the receiving cell. Those reactions often influence which genes become active or inactive. Through this process, cells can influence each other's behavior. A cell might signal its neighbors to divide. Another signal might instruct nearby cells to begin specializing into a particular tissue type. Some signals encourage movement, guiding cells towards specific locations within the developing organism. These exchanges of information create a constantly shifting conversation among cells. One particularly important class of signaling molecules involved in development is known as morphogens. Morphogens are molecules that form concentration gradients across developing tissues. Cells located closer to the source of a morphogen experience higher concentrations. Cells farther away experience lower concentrations. By detecting the strength of the signal, cells can interpret their position within the developing structure. In other words, The amount of morphogen a cell receives acts like a coordinate system telling the cell where it is located. Different concentration levels can activate different genes, leading cells to adopt different identities depending on their position. This mechanism helps explain how tissues organize themselves into precise patterns. A group of cells exposed to high concentrations of a morphogen may become one type of tissue. Cells exposed to moderate concentrations may develop into another type. Cells receiving very little signal may follow an entirely different developmental pathway. Through these gradients, spatial information emerges within the embryo. The concept of morphogens gained strong experimental support through studies of animal development in the late 20th century. Developmental biologists studying fruit flies, amphibians, and vertebrates discovered that certain signaling molecules behave precisely in this way, forming gradients that guide tissue organization. Among the most influential signaling systems identified during this research are several major developmental pathways. One of the best known is the hedgehog signaling pathway. The hedgehog pathway was first discovered through genetic studies of fruit flies. Mutations affecting this pathway produced larvae with unusual patterns of body segments that reminded researchers of a spiky hedgehog. The gene responsible for this pathway was therefore named hedgehog. In vertebrates, related signaling molecules such as sonic hedgehog play essential roles in shaping the developing embryo. Sonic hedgehog helps guide the formation of the neural tube, which eventually develops into the brain and spinal cord. It also influences limb development and the organization of facial structures. Another important communication system is the Notch signaling pathway. The Notch pathway operates through direct contact between neighboring cells. Instead of releasing signaling molecules into the surrounding environment, a cell displays signaling proteins on its surface. When these proteins interact with receptors on adjacent cells, they trigger changes in gene activity within the receiving cell. This type of signaling allows cells that are physically touching each other to coordinate their behavior very precisely. Notch signaling plays an important role in determining cell fate during development. In many tissues, it helps neighboring cells adopt different identities so that tissues form balanced mixtures of cell types. For example, during the development of the nervous system, Notch signaling helps regulate which cells become neurons and which remain as supporting cells. A third major pathway involved in developmental signaling is the W&T pathway. The W&T signaling system was discovered through studies of both fruit flies and mice. The name WNT reflects the combination of two earlier gene names discovered in these organisms. WNT signaling influences many aspects of development, including cell division, cell migration, and tissue organization. In vertebrate embryos, WNT signals help establish the main body axis during early development. Later, WNT signaling contributes to the formation of limbs, organs, and various tissue types. What makes these pathways remarkable is their ability to coordinate the behavior of large groups of cells. Instead of each cell acting independently, signaling pathways allow cells to integrate information from their environment. A cell does not simply ask what genes are present in its DNA. It also asks what signals it's receiving from surrounding cells, what position it occupies within the tissue, and what stage of development the organism has reached. These signals feed into gene regulatory networks that ultimately determine which genes become active. In this way, development emerges from interactions among many cells working together. The genome provides the instructions for producing signaling molecules, receptors, transcription factors, and regulatory proteins. But the final shape of the organism arises through communication and cooperation among millions of individual cells. Cells respond to gradients of morphogens. They exchange signals through pathways such as hedgehog, notch, and windant. They interpret these signals through complex regulatory systems inside the nucleus. And through these countless molecular conversations, tissues begin to organize themselves. Layers of cells fold, stretch, and migrate. Groups of cells begin forming organs, structures emerge, gradually guided not by a single controlling molecule but by networks of interacting signals. The developing embryo therefore behaves less like a machine, assembled piece by piece, and more like a self-organizing system. DNA provides the molecular toolkit, but the architecture of the body arises from collective behavior. Cells constantly communicate, adjust their actions, and respond to their surroundings. Through this ongoing dialogue, the astonishing complexity of a living organism slowly takes shape. As the processes of development unfold, it can sometimes feel as though DNA contains a deliberate plan for constructing living organisms. The sequence of bases appears to contain instructions for building tissues, organizing organs, and guiding the formation of complex biological systems. Yet this apparent design did not arise through intention. Instead, it emerged gradually through one of the most powerful processes known in biology: evolution by natural selection. The idea that living organisms change over time was famously articulated by the naturalist Charles Darwin in the middle of the 19th century. In his book, On the Origin of Species, published in 1859, Darwin proposed that species evolve through the process of natural selection. Natural selection operates through three basic principles. First, individuals within a population vary in their traits. No two organisms are completely identical. Second, some of these variations influence survival and reproduction. Traits that improve an organism's ability to obtain resources, avoid predators, or reproduce successfully tend to increase the likelihood that those traits will be passed to the next generation. Third, these traits can be inherited. Over many generations, beneficial traits become more common within the population. Through this gradual process, organisms become better adapted to their environments. Darwin himself did not know the molecular basis of heredity. The mechanisms of genes and DNA would only be discovered decades later. Yet his theory provided the conceptual framework explaining how complex biological structures could arise without deliberate design. Modern genetics later revealed how variation arises at the molecular level. The DNA sequence within organisms occasionally changes through processes known as mutations. A mutation occurs when the sequence of bases within DNA is altered. This may involve the substitution of one base for another, the insertion of additional bases, or the deletion of a segment of DNA. Mutations can occur for several reasons. Some arise from small errors during DNA replication when cells divide. Others result from environmental influences such as radiation or chemical exposure. Most mutations have little noticeable effect. Some may slightly alter the function of a protein or change how a gene is regulated. A small number can significantly affect an organism's traits. When mutations occur in reproductive cells, they can be passed to the next generation. This continual introduction of variation provides the raw material upon which natural selection acts. Over immense spans of time, natural selection filters these variations. Mutations that disrupt essential biological functions are usually eliminated because the organisms carrying them fail to survive or reproduce effectively. Mutations that improve survival or reproduction may gradually spread through populations. Through countless cycles of mutation and selection, genomes accumulate changes that shape biological form and function. The relationship between genetic change and evolutionary processes became a major focus of 20th century evolutionary genetics. One influential scientist in this field was Moto Kimura, a Japanese population geneticist who introduced the neutral theory of molecular evolution in the 1960s. Kimura proposed that many genetic mutations do not significantly affect an organism's survival or reproductive success. These neutral mutations may spread through populations not because they provide an advantage, but simply through random processes known as genetic drift. According to the neutral theory, a large proportion of molecular changes in DNA accumulate through these random fluctuations rather than through natural selection alone. This insight helped explain why genomes often contain vast numbers of genetic differences that appear to have little functional consequence. Natural selection still plays a crucial role, particularly when mutations influence important biological traits. But Kimura's work highlighted that evolution operates through both selective and neutral processes. The study of evolutionary genetics has also revealed how developmental genes themselves evolve. Genes that control fundamental developmental processes, such as Hox genes or signaling pathways, often remain highly conserved across species. Because these genes play critical roles in building the body plan major, changes to them can be harmful. As a result, natural selection tends to preserve their core functions. Yet evolution still finds ways to modify development without disrupting essential systems. Small changes in regulatory regions of DNA can alter when and where certain genes become active. These regulatory modifications can influence body shape, size, and structural details while preserving the fundamental developmental framework. For example, changes in gene regulation may affect the length of limbs, the arrangement of body segments, or the pattern of pigmentation in animals. Such adjustments allow evolution to explore new forms while maintaining the stability of crucial biological processes. This interplay between conservation and variation has helped generate the extraordinary diversity of life seen across the planet. From insects to birds to mammals, organisms share many of the same fundamental genetic tools. Yet the ways those tools are deployed during development vary widely. The DNA guiding human development therefore represents the result of an extraordinarily long evolutionary history. The earliest self-replicating molecules likely appeared more than 3.5 billion years ago in the ancient oceans of Earth. Over immense stretches of geological time, genetic systems gradually evolved greater complexity. Primitive molecular systems gave rise to simple cells. Cells eventually formed multicellular organisms. Developmental genes emerged that could coordinate the formation of tissues and organs. Through billions of years of evolutionary refinement, these genetic systems became increasingly sophisticated. Each generation carried forward the successful solutions discovered by countless ancestors. When a human embryo develops today, the molecular instructions guiding that process are not new inventions. They represent the accumulated results of an evolutionary journey spanning nearly the entire history of life on Earth. The remarkable ability of DNA to guide the formation of a human body is therefore not the product of foresight or intention. It is the consequence of a long and continuous process of variation, selection, and inheritance. Across unimaginable spans of time, evolution has shaped the genetic instructions that now operate quietly within every developing cell. And through that process, the molecules of heredity have become capable of orchestrating one of the most intricate phenomena in the natural world: the construction of a living organism. Every time a cell divides, it must copy its DNA. Inside the nucleus, The long strands of genetic material unwind so that the molecular machinery responsible for replication can begin its work. Enzymes move along the DNA strands, reading the sequence of bases and assembling new complementary strands to produce two identical copies. This process is astonishingly precise. In human cells, the genome contains roughly 3 billion base pairs. Replicating this enormous sequence accurately requires a complex system of enzymes working together with remarkable reliability. Yet even in the most carefully controlled molecular processes, occasional mistakes occur. These changes in DNA sequence are known as mutations. A mutation simply refers to any alteration in the order of bases within DNA. During replication, the enzyme DNA polymerase reads one strand of DNA and builds the complementary strand. If the wrong base is occasionally inserted, the sequence may change slightly. Mutations can take several forms. One common type is a substitution, in which one base is replaced by another. Another type involves insertions, where extra bases are added into the sequence. Deletions occur when bases are removed. Larger mutations may involve duplications of entire sections of DNA or rearrangements of larger chromosomal regions. Despite the complexity of DNA replication, the error rate is remarkably low. DNA polymerase contains proofreading abilities that help detect mismatched bases during replication. When the enzyme identifies an incorrect pairing, it can remove the misplaced base and replace it with the correct one. Because of these proofreading mechanisms, the probability of an incorrect base being inserted during replication is extremely small. Even so, occasional mutations still occur. From a biological perspective, mutations are not always harmful. In fact, most mutations have little or no noticeable effect on an organism. One reason for this is the structure of the genetic code. The genetic code translates sequences of 3 bases, known as codons, into specific amino acids during protein synthesis. Because multiple codons often correspond to the same amino acid, some mutations do not alter the resulting protein at all. These changes are called silent mutations. Even when a mutation alters an amino acid within a protein, the effect may be minimal. Many proteins can tolerate small changes in their sequence without losing their overall function. Some mutations occur in regions of DNA that do not directly influence protein production or gene regulation. In such cases, the mutation may have no measurable effect at all. For these reasons, a large proportion of mutations are considered neutral. They change the DNA sequence but do not significantly influence the organism's survival or development. Occasionally, however, mutations can affect the function of a protein. If a mutation changes an amino acid critical to the structure or activity of a protein, the protein's behavior may change. Enzymes may become less efficient. Structural proteins may fold incorrectly. Signaling proteins may transmit altered signals. In some cases, these changes disrupt normal cellular processes. Mutations affecting developmental genes can influence how tissues and organs form during embryonic development. Because developmental genes often control large regulatory networks, even small changes may sometimes produce noticeable effects on body structure. Yet it is important to recognize that biological systems possess remarkable resilience. Cells operate within networks of interacting molecules. When one component changes slightly, other pathways may compensate. Multiple genes often contribute to the same biological processes, creating layers of redundancy that help maintain stability. Even at the molecular level, living systems have evolved powerful mechanisms to minimize the impact of genetic errors. One of the most important protective systems involves DNA repair. Cells constantly monitor the integrity of their DNA. Specialized repair enzymes scan the genome searching for damaged or mismatched bases. When damage is detected, repair pathways become activated. One well-studied system is known as mismatch repair. This pathway detects incorrect base pairings that escape the proofreading function of DNA polymerase during replication. Repair proteins recognize the mismatch, remove the incorrect segment, and replace it with the correct sequence. Another pathway called base excision repair corrects small chemical alterations affecting individual bases. Certain environmental influences, such as oxidative reactions inside the cell, can slightly damage DNA bases. Enzymes involved in base excision repair identify these damaged bases and replace them with correct ones. Larger forms of DNA damage are handled by additional repair systems. For example, nucleotide excision repair removes segments of DNA containing bulky distortions, such as those produced by ultraviolet radiation from sunlight. After removing the damaged region, the cell rebuilds the correct sequence using the complementary DNA strand as a template. Even more dramatic repair systems exist to handle double-strand breaks in DNA. Double-strand breaks occur when both strands of the DNA molecule are severed. These breaks can be repaired through mechanisms such as homologous recombination, which uses an undamaged copy of the DNA as a template for repair. Through these multiple layers of protection, cells maintain extraordinary genomic stability. Yet despite these safeguards, some mutations inevitably remain. Rather than viewing these mutations purely as errors, biology recognizes them as part of the dynamic nature of genetic systems. Mutations introduce variation into populations. Variation provides the raw material for evolution. Over long periods of time, this variation allows populations to adapt to changing environments, develop new traits, and explore new biological possibilities. At the level of an individual organism, however, the most striking fact is how reliably development proceeds despite the occasional mutation. A human body forms through trillions of cellular divisions, each involving DNA replication. Even though small errors sometimes occur, the overwhelming majority of developmental processes unfold with remarkable precision. This reliability arises from multiple layers of regulation, repair, and redundancy. DNA replication is highly accurate. Repair systems correct many mistakes before they can accumulate. Gene regulatory networks buffer against small fluctuations in molecular activity. Cells communicate with their neighbors to maintain coordinated development. Together, these systems ensure that biological organisms remain stable and functional, even in the presence of occasional genetic changes. Life therefore operates within a delicate balance. DNA replication strives for accuracy. Repair systems safeguard the genome. Mutations introduce variation. And development proceeds through robust regulatory networks capable of tolerating small imperfections. Through this balance of stability and change, living systems maintain both reliability and the capacity for evolutionary innovation. The genome is therefore not a fragile instruction manual easily disrupted by a single typo. It is a resilient, dynamic system capable of guiding development with remarkable consistency, while still allowing the subtle variations that make biological diversity possible. By the late 20th century, scientists had begun to understand many of the fundamental mechanisms of genetics. Researchers knew that DNA carried hereditary information. They understood how genes could direct the production of proteins. They had uncovered regulatory systems that controlled gene activity during development. Yet an enormous mystery still remained. No one had seen the entire human genome. Scientists knew that the genome must contain vast amounts of information, but its full sequence had never been mapped. The scale of the challenge was immense. Human DNA contains roughly 3 billion base pairs arranged across 23 pairs of chromosomes. If written out as text, the sequence would fill many volumes of printed pages. For decades, geneticists could examine small fragments of DNA, studying individual genes or short sequences. But understanding the entire genome required a project of unprecedented scale. This ambition gave rise to one of the largest scientific collaborations in history, the Human Genome Project. The Human Genome Project began formally in 1990 as an international effort to determine the complete DNA sequence of the human genome. The project brought together scientists, institutions, and research laboratories from several countries, including the United States, the United Kingdom, Japan, France, Germany, and China. The goal was simple in concept, yet enormous in execution: to read every letter of human DNA. Within the United States, a major coordinating role was played by the National Human Genome Research Institute. This organization, part of the National Institutes of Health, helped guide the scientific strategy and support research teams working on genome sequencing. The United States Department of Energy also participated in the project, motivated in part by its earlier interest in understanding genetic damage caused by radiation. At the beginning of the project, DNA sequencing technology was far slower than it is today. Researchers often relied on techniques developed by Frederick Sanger, whose methods allowed DNA fragments to be sequenced by identifying the order of bases. Using these methods, scientists could determine sequences of several hundred bases at a time. But sequencing 3 billion bases required dividing the genome into countless smaller fragments, sequencing each fragment individually, and then assembling the pieces together like a vast molecular puzzle. Large sequencing centers were established in research institutions around the world. Teams of scientists, technicians, and computational specialists worked together to produce and analyze DNA sequences. Powerful computer algorithms were required to reconstruct the genome from overlapping fragments. As sequencing progressed through the 1990s and early 2000s, the project gradually revealed the structure of the human genome in increasing detail. In the year 2000, scientists announced the completion of a preliminary draft sequence of the human genome. A more refined and highly accurate version followed several years later. The accomplishment represented a milestone in modern science. For the first time, researchers could examine the complete sequence of human DNA. Yet one of the most surprising discoveries that emerged from the Human Genome Project concerned the number of genes within the genome. Before sequencing began, many scientists had predicted that humans might possess 100,000 or more genes. After all, humans are complex organisms with intricate organs, nervous systems, and cognitive abilities. But when the genome was analyzed, the number of protein-coding genes turned out to be much smaller. Current estimates suggest that humans possess roughly 20,000 protein-coding genes. This number is only modestly larger than the number of genes found in many other animals. At first glance, this result seemed puzzling. How could such a relatively modest number of genes produce the extraordinary complexity of the human body? Part of the answer lies in the versatility of gene regulation and protein function. Many genes can produce multiple forms of proteins through processes such as alternative splicing. In this process, different segments of RNA transcripts are combined in different ways, allowing a single gene to generate several related proteins. Another important insight from the Human Genome Project involved the overall composition of the genome. Only a small portion of human DNA directly codes for proteins. In fact, less than 2% of the genome consists of protein coding sequences. The remaining majority of DNA includes regulatory elements, repeated sequences, structural components of chromosomes, and regions whose functions are still being investigated. For many years, some scientists referred to non-coding DNA as junk DNA, assuming that it served little purpose. However, research over the past several decades has revealed that many of these non-coding regions play crucial roles in regulating gene activity. Regulatory DNA sequences control when and where genes become active. Some regulatory elements function as promoters located near the start of genes. Others act as enhancers, which can influence gene activity from much greater distances along the chromosome. Enhancers bind transcription factors that help activate or suppress gene expression. Through these interactions, regulatory DNA sequences help determine which genes operate within specific tissues and at particular moments during development. This regulatory system allows a relatively modest number of genes to generate enormous biological complexity. Genes can be activated in different combinations across different cell types. They can turn on and off at specific stages of development. They can respond to signals from neighboring cells or environmental influences. The genome therefore functions not simply as a list of genes, but as a highly dynamic regulatory landscape. The Human Genome Project also stimulated rapid advances in sequencing technology. New methods known as next-generation sequencing dramatically increased the speed at which DNA could be analyzed. Sequencing that once required years can now be performed in days or even hours. These technologies have transformed genetics, medicine, and biological research. Scientists can now compare genomes across species to study evolution. Physicians can examine genetic variants associated with disease. Researchers can investigate how regulatory DNA influences gene activity during development. The ability to read the human genome has therefore opened new frontiers in understanding how DNA guides the formation and function of living organisms. And yet, even with the complete genome sequence available, the central mystery remains deeply fascinating. The genome contains billions of letters. Hidden within those letters are instructions capable of guiding the development of a human body from a single fertilized cell. Deciphering how those instructions operate remains one of the most profound scientific challenges of the modern era. The Human Genome Project provided the map. But understanding how the map is interpreted during development continues to reveal new layers of biological complexity. Over the past century, scientists have gradually uncovered many of the mechanisms through which DNA guides the development of living organisms. They have identified genes, mapped genomes, studied regulatory networks, and observed the molecular machinery operating inside cells. Yet biology remains a science of discovery. Each generation of researchers develops new tools that allow them to look deeper into the processes of life. With every technological advance, previously hidden layers of genetic regulation begin to emerge. Modern biology now possesses an extraordinary collection of techniques for studying genes. These tools allow scientists not only to observe genetic activity, but also, in some cases, to alter it deliberately in order to understand how genes influence development. One of the most transformative technologies developed in recent years is a gene editing method known as CRISPR. The name CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. These repeating DNA sequences were first discovered in bacteria during studies of microbial genetics. At first, the function of these unusual sequences remained unclear. Over time, researchers discovered that they form part of a bacterial immune system that helps bacteria defend themselves against viruses. When a virus infects a bacterial cell, Fragments of the viral DNA can be captured and inserted into the bacterial genome within these repeating sequences. The bacterium then uses these stored fragments to recognize and destroy matching viral DNA during future infections. This system relies on specialized enzymes that can locate specific DNA sequences and cut them with remarkable precision. Two scientists played especially important roles in transforming this natural bacterial defense system into a powerful tool for genetic research. Jennifer Doudna at the University of California, Berkeley, and Emmanuelle Charpentier, who conducted research in several European institutions including the Max Planck Unit for the Science of Pathogens, worked together to understand how the bacterial CRISPR system functions. In the early 2010s, their research revealed how the enzyme Cas9 could be guided by a small RNA molecule to locate a specific DNA sequence. Once the enzyme reaches its target, it cuts the DNA at that precise location. This discovery opened the door to a new approach to gene editing. Scientists could design RNA molecules that guide the Cas9 enzyme to almost any location in the genome. Once the DNA is cut, cellular repair systems attempt to repair the break. During this repair process, researchers can introduce small changes to the DNA sequence, effectively editing the gene. The CRISPR system therefore allows scientists to alter genes with extraordinary precision. Researchers can disable genes to study their function. They can introduce new sequences to explore how genetic changes influence development. They can even correct certain genetic mutations within cells. For their groundbreaking work on this technology, Jennifer Doudna and Emmanuelle Charpentier were awarded the Nobel Prize in Chemistry. While gene editing has captured widespread attention, many other tools also play essential roles in genetic research. One of the most important approaches involves the use of model organisms. Model organisms are species chosen for scientific study because they are relatively easy to maintain in laboratories and possess genetic systems that resemble those of more complex organisms. Several model organisms have become particularly important in developmental biology. The fruit fly species Drosophila melanogaster has been used for over a century to study genetics. Its short life cycle and well-understood genome allow scientists to investigate how specific genes influence development. Fruit fly embryos develop rapidly, enabling researchers to observe developmental processes over relatively short periods. The house mouse, Mus musculus, serves as another major model organism. Mice share a large portion of their genes with humans and possess similar physiological systems. Genetic techniques allow researchers to create mice in which specific genes are altered or removed. Observing how these changes affect development helps scientists understand the roles of those genes. Zebrafish have also become widely used in developmental research. Their embryos develop outside the body and remain transparent during early stages. This transparency allows researchers to observe cell division, tissue formation, and gene activity under the microscope in living embryos. Through studies of these model organisms, scientists can investigate genetic mechanisms that would be difficult or impossible to examine directly in humans. Another set of powerful tools involves advanced microscopy. Modern imaging technologies allow researchers to observe biological processes at extraordinary levels of detail. Fluorescent proteins can be attached to specific molecules inside cells, causing those molecules to glow under particular wavelengths of light. Using these fluorescent markers, scientists can track the movement of proteins, observe the activation of genes, and follow the behavior of cells during development. Confocal microscopy and related imaging techniques can produce detailed three-dimensional images of tissues, revealing how cells organize themselves within developing organs. Even more advanced methods allow researchers to observe individual molecules interacting within living cells. At the same time, advances in DNA sequencing technology have dramatically expanded the ability to study gene activity. Earlier sequencing methods focused primarily on determining the order of bases in DNA. Modern approaches now allow scientists to measure which genes are active within specific cells at particular moments. Techniques such as RNA sequencing reveal the collection of messenger RNA molecules present in a cell. Because messenger RNA reflects which genes are currently being expressed, this method allows researchers to examine patterns of gene activity across tissues. More recent approaches, known as single-cell sequencing, allow scientists to study gene expression within individual cells rather than averaging signals across large populations. These techniques have revealed that even cells within the same tissue can exhibit subtle differences in gene activity. By combining advanced sequencing technologies with imaging methods, researchers can build detailed maps showing how genes operate during development. Scientists can observe where genes activate, when they activate, and how their activity spreads across tissues. These insights are helping reveal previously hidden layers of genetic regulation. Gene regulatory networks appear even more intricate than once imagined. Epigenetic modifications interact with transcription factors. Cell signaling pathways influence gene activity. Chromatin structure shapes which regions of DNA remain accessible. Through modern technologies, scientists can now explore these interactions with unprecedented clarity. Yet the deeper researchers look, the more complexity they uncover. The genome does not operate as a simple linear instruction manual. Instead, it functions as a dynamic system of interacting molecules, regulatory networks, and signaling pathways. Modern genetic tools have therefore transformed the study of biology from a discipline focused primarily on individual genes into one concerned with entire systems of regulation. These technologies continue to reveal new layers of organization inside living cells. And with every discovery, the original question remains as fascinating as ever: how a sequence of molecules inside a cell can help guide the formation of an entire living organism. As scientists have explored the mechanisms of genetics and development, one realization has gradually become clearer. Living systems do not behave like simple machines following a rigid set of instructions. Instead, they behave more like complex networks of interacting parts. The DNA inside a cell certainly contains important information, but the behavior of a living organism cannot be understood by reading the genetic sequence alone. Genes interact with one another. Proteins influence other proteins. Cells respond to signals from neighboring cells. Entire tissues exchange chemical messages as development unfolds. What emerges from these countless interactions is something greater than the individual components. This phenomenon is often described as emergence. An emergent property is a feature of a system that arises from interactions among its parts, rather than from any single component acting alone. In biology, many of the characteristics associated with life itself emerge from networks of interacting molecules and cells. For example, no single gene contains the complete instructions for building a heart. Instead, the heart forms through coordinated interactions among many genes, proteins, and signaling pathways. Cells communicate with one another, respond to chemical gradients, and organize themselves into tissues that eventually form a functioning organ. The structure of the heart emerges from these collective interactions. One scientist who explored these ideas in depth was the theoretical biologist Stuart Kauffman. Kauffman studied how complex systems behave when many components interact with one another. His work suggested that biological systems possess a natural tendency towards self-organization. Self-organization refers to the spontaneous emergence of order within complex systems without requiring an external designer controlling every detail. Kauffman explored how networks of genes might behave when they regulate one another through feedback loops. In his theoretical models, genes could activate or suppress other genes, creating networks of regulatory interactions. When large numbers of these interactions occur simultaneously, the network can settle into stable patterns of activity. These patterns may correspond to different cell types. For example, one stable pattern of gene activity might produce the gene expression profile of a neuron. Another pattern might correspond to a muscle cell. A third might represent a skin cell. The important point is that these patterns arise from the network itself. They do not require an explicit blueprint describing every molecular step. In this view, the genome functions less like a detailed architectural plan and more like a set of interacting components capable of generating organized behavior. These ideas have influenced the development of a field known as systems biology. Systems biology attempts to understand living organisms by studying the networks of interactions that connect genes, proteins, metabolites, and cells. Instead of focusing on individual genes in isolation, systems biology examines how entire networks behave collectively. Researchers in this field use mathematical models, computational simulations, and large-scale experimental data to map biological networks. These networks include gene regulatory networks controlling gene expression, protein interaction networks connecting enzymes and signaling molecules, and metabolic networks governing chemical reactions within cells. Through these approaches, scientists have discovered that biological systems often display properties similar to other complex systems found in nature. Networks of interacting elements can produce stable states, oscillations, waves of activity, and coordinated behavior across large numbers of components. Such behaviors are not programmed explicitly in any single gene. They emerge from the interactions within the network. An example can be found in developmental pattern formation. When groups of cells communicate through signaling molecules, they can spontaneously organize into patterns such as stripes, segments, or repeating structures. These patterns arise because cells respond to signals from their neighbors while producing signals of their own. Through feedback and interaction, ordered structures appear across the tissue. Similar principles operate at many levels of biological organization. Proteins interact with other proteins to form molecular complexes. Cells communicate with neighboring cells to form tissues. Tissues integrate into organs that perform coordinated functions. Each level of organization builds upon interactions occurring at the levels below. Understanding life, therefore, requires examining how these layers of interaction operate together. This perspective helps explain why the human genome alone cannot fully describe the complexity of the human body. The genome provides the molecular components and regulatory instructions, but the behavior of the system emerges through interactions among those components. Proteins fold into structures that allow them to interact with other molecules. Gene regulatory networks control patterns of gene expression. Cell signaling pathways coordinate behavior across tissues. Physical forces influence how cells move and organize during development. All of these processes interact simultaneously. In this sense, development resembles a vast conversation taking place among trillions of cells. Each cell responds to signals, sends signals of its own, and adjusts its behavior according to the changing environment. Through these ongoing interactions, order gradually appears, organs form, tissues organize, structures emerge that allow the organism to survive and function. The remarkable outcome is that complex biological organization arises without requiring any single molecule to understand the entire process. DNA does not contain a miniature model of the body. Instead, it provides the molecular ingredients and regulatory systems that allow development to unfold through dynamic interactions. Life therefore behaves as a complex system. Genes, proteins, cells, and tissues participate in networks that generate structure and function through cooperation and feedback. From these interacting systems emerges the astonishing phenomenon of a living organism developing from a single cell. The more scientists study these networks, the clearer it becomes that biological complexity arises not from simple instructions, but from the rich interplay of countless interacting parts. By this point in the journey, it may almost seem as if DNA possesses a kind of quiet intelligence. Inside nearly every cell of the body sits a molecule containing billions of chemical units arranged in a precise sequence. From that sequence emerges the development of tissues, organs, and entire biological systems. Cells read those sequences, respond to them, and coordinate their actions with astonishing reliability. It is therefore tempting to speak about DNA as though it somehow knows how to build a human body. Yet that description, while convenient in everyday language, hides an important truth. DNA does not actually know anything. A molecule cannot possess knowledge, intention, or awareness. DNA is composed of atoms arranged into chemical structures. Those atoms obey the same physical laws that govern every other part of the natural world. What DNA contains is not knowledge, but information encoded through chemistry. The sequence of bases within DNA determines which proteins can be produced inside the cell. Proteins then perform a vast range of functions. Some act as enzymes that accelerate chemical reactions. Others form structural components of tissues. Many serve as regulatory molecules controlling when and where genes become active. Through these molecular interactions, cells can interpret the information stored within DNA. The instructions themselves arise from the order of the bases. Adenine tends to pair with thymine. Cytosine tends to pair with guanine. These pairing rules allow DNA to be copied during cell division with remarkable accuracy. They also allow RNA molecules to read the DNA sequence and translate that sequence into proteins through the genetic code. All of this occurs through chemistry. Enzymes bind to DNA because their shapes match specific molecular structures. Transcription factors recognize certain sequences because chemical interactions allow them to attach at those locations. Ribosomes assemble amino acids into proteins because messenger RNA carries sequences that specify which amino acids should be joined together. At every stage, the system operates through physical interactions between molecules. No molecule is aware of the final outcome. Yet from these countless molecular interactions emerges a process capable of building an entire organism. The reason this process works so reliably lies in the long history of evolution. Over billions of years, genetic systems have been shaped by natural selection. Mutations occasionally altered DNA sequences, sometimes changing how genes were regulated or how proteins functioned. When those changes improved survival or reproduction, they tended to persist within populations. Over immense spans of time, these small adjustments accumulated. Genes that contributed to reliable development were preserved. Regulatory systems that coordinated cellular behavior were refined. Molecular mechanisms that protected the genome from damage became more sophisticated. Each generation inherited genetic systems shaped by the success of previous generations. The result is a genome containing instructions that function with extraordinary precision. Yet even with this precision, development does not proceed according to a rigid script. The genome provides molecular instructions, but the outcome emerges through interactions among genes, proteins, cells, and physical forces within the embryo. Cells communicate with one another through signaling molecules. Gene regulatory networks coordinate patterns of gene activity. Epigenetic mechanisms influence which regions of DNA remain accessible. These interacting systems allow development to adapt and stabilize as it unfolds. In many ways, the process resembles a complex conversation taking place across trillions of cells. Each cell interprets signals from its environment. It responds by activating certain genes and suppressing others. Those genetic changes influence the proteins produced within the cell, which in turn affect how the cell behaves. Through this ongoing exchange of signals, tissues gradually organize themselves. Structures emerge not because a single molecule directs every detail, but because many molecular systems interact according to consistent physical rules. One might compare this process to patterns forming in other complex systems found in nature. Snowflakes form intricate structures as water molecules crystallize according to simple physical rules. Spiral galaxies emerge as gravitational interactions shape the motion of stars. Sand dunes arrange themselves into repeating patterns as wind moves grains across a desert. In each case, large-scale structure emerges from many small interactions. Biological development follows a similar principle. Chemical rules govern how molecules interact. Regulatory networks coordinate gene activity. Cells respond to local signals and physical forces. From these interactions arises the organized complexity of a living organism. The human body contains trillions of cells, organized into tissues, organs, and systems capable of performing extraordinarily sophisticated functions, yet every one of those cells originated from a single fertilized egg. That transformation may appear almost miraculous, but when examined closely, it emerges from processes that are entirely consistent with the laws of chemistry and physics. DNA does not know how to build a human being. Instead, the sequence of DNA provides the molecular framework through which cells can interact, regulate their behavior, and respond to signals during development. Those interactions, shaped by billions of years of evolution, allow biological systems to generate order and structure from molecular activity. The remarkable truth is that something as simple as a chemical sequence can participate in processes capable of producing life. From the pairing of nucleotides to the folding of proteins, from the communication between cells to the emergence of tissues and organs, complexity gradually unfolds from relatively simple beginnings. And perhaps that is the most fascinating aspect of all. Life does not arise from mysterious forces hidden beyond the laws of nature. It arises from the patient accumulation of molecular interactions operating over immense spans of time. Within every cell, DNA quietly participates in this ancient process. A molecule carrying information shaped by the deep history of life on Earth. A sequence of chemical letters that, through interaction and evolution, helps guide the formation of a living human being. After exploring the many layers of biology that guide development, it is natural to return to the original question. How does DNA know how to build you? At first glance, the idea seems almost mysterious. Inside nearly every cell of the human body lies a molecule containing billions of chemical units arranged in a particular order. From that sequence emerges a process capable of guiding the development of an entire organism. A fertilized egg divides, cells specialize, tissues organize, and organs gradually appear. Bones grow, muscles form, nerves extend through the body, and the brain develops networks of astonishing complexity. All of this unfolds from a molecular beginning so small it could easily rest on the tip of a pin. Yet the journey through genetics and development reveals that this apparent mystery is not the result of hidden knowledge inside the molecule itself. DNA does not possess awareness or intention. Instead, it provides information encoded through chemistry. The sequence of bases within DNA influences which proteins can be produced inside a cell. Those proteins act as enzymes, structural components, signaling molecules, and regulatory factors that control countless biochemical processes. Through these molecular activities, cells are able to read the information stored within the genome. But the genome does not operate alone. Gene regulatory networks coordinate which genes become active in particular cells. Epigenetic mechanisms influence which regions of DNA remain accessible. Signaling molecules allow cells to communicate with their neighbors. Morphogen gradients help establish positional information during early development. Through these interacting systems, cells gradually organize themselves into tissues and organs. The process unfolds across many layers of regulation. Genes encode proteins. Proteins influence cellular behavior. Cells communicate through chemical signals. Tissues form through collective interactions among many cells. Each layer builds upon the layers below it, producing a system capable of generating remarkable biological complexity. Yet even this description only explains part of the story. Behind the functioning of the genome lies an even deeper history. The genetic systems operating inside modern organisms did not appear suddenly. They are the result of an evolutionary process stretching back billions of years. Across unimaginable spans of time, mutations introduced variation into ancestral genomes. Natural selection favored genetic systems capable of supporting reliable development and successful reproduction. Gradually, molecular mechanisms evolved that could coordinate increasingly complex biological structures. Gene regulatory networks emerged. Signaling pathways refined communication between cells. Epigenetic systems help stabilize patterns of gene expression. Through countless generations, these systems were refined by evolutionary pressures. The DNA sequence carried by every organism today represents the accumulated result of this long evolutionary history. Each genome contains solutions discovered by many generations of life that came before. When a new organism develops, it is therefore participating in a process shaped by deep time. The molecular interactions occurring inside each cell follow patterns that have been tested repeatedly across the history of life on Earth. In this sense, development is not simply the execution of a genetic program. It is the unfolding of a complex system that has been shaped and refined over billions of years. From the perspective of chemistry, the process is composed of countless molecular interactions. Enzymes bind to DNA. Proteins fold into functional structures. Cells exchange signals with their neighbors. From the perspective of biology, those interactions produce organized tissues, functioning organs, and living organisms capable of sensing and responding to the world. And from a broader perspective, each living organism represents the continuation of an ancient lineage reaching back through evolutionary history. The remarkable outcome is that something as small as a sequence of molecules can participate in processes capable of building life. Inside every cell, DNA quietly interacts with thousands of other molecules. Proteins fold, signals travel between cells, and regulatory networks coordinate patterns of activity across tissues. These processes never truly stop. Even at this moment, countless molecular events are unfolding inside the body. Cells are dividing, repairing themselves, responding to signals and maintaining the intricate systems that sustain life. All of this activity traces back to the genetic information stored within DNA and the molecular networks that interpret it. The next time the idea arises that DNA somehow knows how to build a human being, it may help to remember what is truly happening. A sequence of chemical letters shaped by billions of years of evolution is interacting with an extraordinary network of molecules inside every cell. From those interactions emerges the living organism, quietly, continuously, and with a complexity that still inspires deep scientific curiosity. Within the microscopic world of cells, life is always in the process of building itself,